Hello everyone, and welcome to the second video in this lecture series on building protein transformers from scratch. So in this video, we're going to take the data set that we constructed in the first video, and we're going to model it using a very simple protein language model. And in particular, we are going to use the architecture from this paper, this 2003 paper. So this is the same paper that's used in Andre Karpathy's Neural Network Zero to Hero course. And while the data that we're modeling in this course, protein sequences, is quite different than the data that we model in that course, the underlying modeling architecture is surprisingly similar. And we're going to actually be able to implement this paper sort of almost word for word on our protein data and have it work sort of fairly well for a 2003 architecture. So let's dive into building a very simple protein language model. So we'll again use a Jupyter Notebook here. And so these are our imports from last time, Plotly, Express, and Biotite. To these imports, I'm going to add two more for this lecture. So in particular, we're going to use Torch, and we're also going to use Rich. Rich is just a fun library for pretty printing in Python. And it will, in this case, we're using it for a progress bar. So we'll import these packages and we'll dive in. So the first thing we're going to do is obviously read our data into this notebook. And so we're going to do this the same way that we did previously by using Biotite to read the FASTA file, extracting the protein sequences. And in this case, let's just verify that the maximum protein length that we observe is what we expect. In the previous video, we restricted these lengths to be less than 128. As we can see, the longest one in this data set is 127. And these are these are the first three examples. So that's the data we're trying to model. And in the NLP context, if we go back to the paper, I don't know if this is the exact paper that introduced the idea of word embeddings. Maybe it was a slightly earlier paper by the same group. But this paper uses word embeddings in the following way. So rather than representing each word as an integer or one hot or some other thing, each word is assigned a vector, a dense vector. And those vectors are all stacked up into this embedding matrix that they call C. And in our case, we're going to use the exact same architecture. So we're going to represent each protein letter as an integer, and then we're going to use that integer to index into this embedding table in the same way that they do in this paper here. So in order to do this, we need to assign a unique integer to each amino acid and build a mapping from one to the other, otherwise known as a tokenizer. And so this is the code. This is some code that'll do that for us. So this just prints out the number of examples in our data set, the maximum length here, the number of unique characters in the vocabulary, which is the 20 amino acids. And then here is the vocabulary itself. So there's nothing weird in here. We've already excluded ambiguous characters X, and it's just the 20 amino acids. And so now we need to build like a tokenizer object that we can actually use to map sequences from the string representation to the integer representation and back and forth. And so we're just going to use a simple Python mapping for this. We call this S2I string to index. And in particular here, we're going to use the character dot as a special character that represents sort of padding or no protein sequence. And we're going to use this later on. So we'll assign this to 0, and then we'll start at 1 for our the rest of the amino acids. And we can see that we'll print the reverse mapping here. Every amino acid is assigned to an integer and vice versa, with the special character dot represented by 0. Okay, so now that we have a mapping, we need to transform our data set from a list of strings into this list of padded integers. And so this code from the Neural Network Zero to Hero series, I think does a great job of showing just a very clean way to do this. So the first thing we need to introduce is this concept of the block size. So in this paper, they've used sort of 3 characters to predict the next character in the sequence, and that's the context that they're modeling. And so in our case, we're going to do the same thing. We're going to use a block size of 3, so we're going to use 3 amino acids to predict the next amino acid. So this little block of code here builds up the data set X, which is all the things that the model is going to see, and then Y is all the predictions that we hope it's going to be able to make. So what we do is we loop over each example, and we build up examples, or we build up training examples where we have 3 amino acids in one list, and then the 4th in a separate list. And let's just run this code and print out, just in particular, what this actually looks like. It illustrates that in a natural language modeling context, for every training example, which is like a long sequence, we actually have many training examples sort of packed into it because we're only predicting one character from the previous 3. So what we can kind of do is kind of slide across the sequence that we're trying to model and assign training examples all along the sequence. So in particular here, you can see that the first training example is just a blank context. And for that, we want the model to always predict methionine, the first residue in the protein. And then as we go into the protein here, the model will receive this sequence of amino acids, cysteine, leucine, leucine, and we want it to predict serine there. And so all of our data set, which has 27,000 protein sequences, we actually have many more training examples because we're only looking at 3 characters at a time. And in particular, we have over 2 million training examples once we break the data set up in this way. As is totally standard, we're going to split the data set into training portion, a test portion, and a validation portion. And we're going to do this with 90% of the data in the training set, 10% in the validation set, and 10% in the test set. And so this code is just a clean way to do this. We generate a random permutation of the integers in the length of our data set. And then we look at sort of the first 90%, the next 10%, and then the last 10% to form our different data sets, the training set, the validation set, and the test set. And then we can just print out the shapes of these. So we can see in our training set, we have just over 2 million sequences. In the validation set and the test set, we have about 250,000 each. Okay, so now that we have our data represented as training examples, and again, these are 3 integers followed by the 4th integer, which is trying to be predicted, we can build our model. So if we go back to the paper, so we've just implemented this part, just the index for each character. So now we need our lookup table. So again, this is simply a matrix that has a number of rows equal to the vocab size and columns equal to the embedding size, or the size of each embedding vector. And we can see in PyTorch, indexing into this embedding table is really easy. We can actually just index with the X object directly and get embeddings for all the amino acids in the data set at once. So in particular here, when we index into C, we receive a stacked up tensor. So this is all the training examples. And then for each training example, we have 3 individual amino acids. And for each one of those, we have a 10-dimensional embedding vector. So that's the shape of this. So the next step in the network is this linear layer here with a TANH activation at the end. So we know how to build this in PyTorch. Linear layer is a weight matrix and a bias vector. And so in particular, the weight matrix in our case, it needs to accept 3 amino acids. So it needs to be 3 times the embedding size, so 30 in our case. We're going to somewhat arbitrarily use a hidden size of 100 here. for the weight and for this layer. And we'll just look at sort of what is the hidden state after this layer, after we do a forward pass through this layer. So in particular, we take our embedding and we reshape it. Here we have the stacked up. So there's 3 embedding vectors stacked up together. Here we're going to unroll those and put them into one long vector of length 30. We're going to multiply that by the weight matrix and then add the bias, and that will produce like the first hidden state of our network. And then we'll do the output layer. The output layer maps from the hidden size to the vocab size, and it's the same weight matrix and bias vector. And then to get the logits, we simply multiply our hidden state by the second weight matrix and add the second bias vector. And we can see that the logits are over the vocabulary size, or the 20 amino acids. Then we can get unnormalized counts and probabilities from the logits. And we can see that now these probabilities, this all sums to 1. And let's just evaluate quickly the loss of this untrained model on our data set using the negative log likelihood loss. This is the same as the cross entropy we'll be using later, except we've manually done all the steps here that cross entropy does for us. So as you'd expect, this loss is quite high. We've not trained the model yet. Okay, so let's just put this in a more cleaned up form. And we can abstract a few of these parameters away as variables in case we want to change them later. But this is the basic outline of the network. We can see the output here is the probabilities over all the different tokens in the data set. And so here in the cell 15, we'll establish the block size, embed size as variables, as well as the hidden size. And then we can kind of rebuild the network nicely and cleanly in this one cell. All right, so let's look at the number of trainable parameters in our model. This model has just over 10,000 trainable parameters, so it's a very small model. Let's see if we can actually train this on some of our sequences in our data set. Okay, so here's the training loop we're going to use. So we're going to keep track of the training loss throughout training. Let's train for 200,000 steps. And so for each step, what are we going to do? There's a really small number of things we need to do at each step. First is we're going to choose a mini batch to operate on. The way we're going to do this is pick 32 random integers from our data set and pull those out. We're going to embed them using the embedding table C, which will give us a tensor of size 32 by 3 by 10. Again, this is 32 training examples. Each training example has 3 amino acids, which are represented by vectors of length 10, and it's all stacked up together. To do a forward pass through the model, we flatten this out. Then we multiply it by the weight matrix, add the bias, and then put the whole thing through the tanh activation. The result of this is a hidden layer for each sequence in the batch. We get the logits by multiplying with the output layer, adding the output bias. And then finally, we can calculate the loss using cross entropy. So cross entropy is a much more numerically stable version of what we did up here. So cross entropy takes the logits directly, and it will do this, get the counts and probabilities and stuff for you, and it'll do it in a numerically stable way. So it's recommended to use that. Once we have the loss, we can do the backward pass, set all the gradients to none, calculate the gradients with backward, and then we can update the network using this learning rate and what I've done here is the learning rate will be 0.1 for the first half of training. For the second half of training, it'll go down to 0.01. Again, this follows neural network 0 to hero parameters pretty closely. And then finally, on each step of the training, we'll keep track of the training loss, and then so we can print that after. So let's run this. And so this is a rich progress bar. And so as this is training, let's just briefly review what we've done so far in this notebook. So in particular, we have read in our data set that we've previously filtered. We have counted up the number of characters and established a vocabulary. We have built a tokenizer, a character level tokenizer that represents our sequences as numbers for a language model. We've built a data set using a block size or a context length of 3. So for every sequence, we pull out all these different examples where we have 3 amino acids, and we're predicting the 4th one. We stack these up into nice torch tensors. We shuffle and split these different tensors into a training set, a validation set, and a test set. And then we build out a very simple natural language model based on this paper. Let's see how the training has gone here. Is done. And we've achieved a loss of 2.47. And let's just look at this loss over the mini batches. So it looks like we have a classic sort of hockey stick feature here, indicating our model is not particularly well initialized at this stage. We can fix that later. But it does look like the model has learned something, evidenced by the training loss going down. So one important feature of the model that we're implementing here is that each individual token is represented by a dense vector. And why is that important? It's because this allows the model to learn things like 2 words are very similar. And you can imagine that if the model is training and it sees that 2 words are often interchangeable in a sentence, the model can assign embedding vectors that are close together in embedding space, representing the similarity between those 2 words. And in training a protein language model, we want pretty much the same thing to be true. We know that different amino acids are more similar to each other than others. And we want the model to be able to recapitulate this. We want the model to learn about the similarities between the different amino acids. And one way to look at and see and evaluate if your model is any good is let's visualize the embedding space that the model has learned. And so in particular, to do this, we'll plot the first 2 dimensions of the embedding matrix in a 2D plot, and then overlay them with the different amino acids. And so if you're looking in a plot like this, see if you can convince yourself that the model has learned that similar amino acids are similar in this plot, or close together in this space. So you might be able to convince yourself that here, you have all of the isoleucine, valine, leucine, methionine. So you have sort of aliphatic amino acids. Maybe over here, you have some charged grouping together. Clearly cysteines is off on its own. Maybe you can convince yourself that there's some structure in this plot that represents, or that gives you some indication that the model has learned that different amino acids are different. I don't know if you'd expect to see something super clear, just the level of training we've done so far. But this is definitely one really useful thing to think about when you're training these models. Thinking about how to automate this process, we'll talk about that in lecture 4. And as a final thing, I think it would be just interesting to see what kind of samples this model generates. So in order to do that, we'll use this sampling procedure that is now sort of classic in NLP. So what we do is we start with a context that's empty, so it's all zeros. We pass that context through the model. We embed it, we get the hidden vector, we get the logits, the probabilities here directly with softmax, and then we sample from those probabilities using torch multinomial to give us sort of the next amino acid in the sequence. And we do this autoregressively to build up a new sequence. We'll do this 20 times, and we'll just look at the 20 sequences that result. So looking at these sequences that our trained model has produced, they look pretty interesting. So some of them are obviously too short, probably not a real protein. Some of them are maybe sort of the right length. Here, let's look at this one. And so this is a really interesting aspect of training protein language models that's dissimilar from training from training natural language models. So in this paper and in the Neural Network Zero to Hero series, when the model prints out the names that's generated, you can just sort of look at the names and figure out, is the model any good at producing names? Because your human machine, your brain, is extremely good at processing text and can do all this stuff. However, when you're training a model like this one that produces protein sequences, even somebody like me who has a PhD in designing proteins and has been designing proteins professionally now for like a decade, I can tell you very, very little about these sequences just looking at them with my eyes. I can tell you this one's probably not an enzyme, that's just because it's short. This one, if it's the right length, it's very hard for me to tell anything about this just by looking at it. Maybe if I looked at it and stared at it and kind of imagined it in my brain. And so this is just a fundamental difference between the field of natural language processing and the field of modeling protein sequences. Even though we're using the same technology, we're using natural language models, language processing, transformer models, the evaluation is fundamentally more difficult in a biological context. If you really want to know if a name is a good name when it comes out of your model, you can just look at it and say, that's a good name. If you really want to know if this protein sequence is a real protein, or if it folds, or if it functions, there are a lot of steps that you have to do with it. You have to take this protein sequence, and using the DNA sequence, you have to design, using the protein sequence, you have to design a DNA sequence that will code for this. That will, when it's transcribed, it'll make an mRNA that translates to this protein sequence. You have to do that in the context of a specific organism. There is no generic biological compiler or runner, right? It has to be in a specific living organism. And then once the living organism produces your protein, you have to purify the protein away, and you have to put it into a biochemical assay of some kind. It would be an understatement to say that this is a very complicated process compared to just looking at a piece of text. And so this is just sort of an interesting wrinkle in the field of using natural language processing techniques for modeling proteins. In silico, there's very little we can do. I'll take this and I'll put this in the ESM Metagenomic Atlas, and we'll fold it with ESM fold. This is one thing that you can do with generated sequences. So let's put this in, and let's see if ESM fold comes up with a plausible structure for this. If ESM fold comes up with a plausible structure, we think maybe it's a good protein. If it comes up with, this, I don't know. Surprisingly, this has some of the features of the protein we're seeking to model. So the protein we're seeking to model has some beta sheets, and it has 2 alpha helices. Surprisingly, ESM fold is predicting in this sequence sort of similar topology, but everything else is wrong. Based on this prediction, there's no way this is going to really fold and function. And so it tells us that the neural network model that we've built here is just too simple for this problem. It's not surprising. This is architecture that came out in 2003. It's 20 years old now. And obviously, modeling protein sequence is more complicated than modeling short names. In the next lecture video, lecture 3, we're going to expand this model to a transformer architecture. And the transformer architecture takes into account the whole sequence, among other things, and we hope that that architecture can generate much more realistic proteins by modeling our data distribution much better. So we'll see you in the next video.